Okay, before we begin, are there any questions? Can somebody confirm you can hear me?
We can hear you.
Great. Let me get to where we are. There we are. Let me share my screen. Should be seeing my screen now. Not sure what that's going up. All right, today is March 12th, and we are supposed to be starting chapter 7. We need to finish up chapter 13. Any question about that? In the lab today, I'm going to be logging in, but only for 15 minutes to answer questions. After that, I'll log out. Let me remind you that on Thursday, we have a review for, I thought I put that on there. We have a review for the final in the lab. Any question about that? Hopefully on Thursday, well, actually, hopefully today we'll. I suppose I could put the review on Thursday. I didn't think of that. Well, let's see where we go, and then I'll consider when we'll have the review. But I'm thinking of having it this Thursday in the lab, but it would be better if we could have it on Thursday in the lecture. And it looks like I don't have anything scheduled for Thursday on the lecture, although chapter 7 is a little longer than one day, but not much longer. All right, let's begin with chapter 13. We had talked about the T-even viruses, talking about how T4 is shaped like the lunar lander. And T4 is the virus that has been studied more than any other virus. When an animal gets a viral infection, the host immune system will respond to it. Infection with the virus stimulates the host to produce antibodies. The antibodies recognize and bind to the viral surface proteins. And these are the antigens that we discussed last time. Some viral proteins can change or mutate frequently. I thought we talked about this, but maybe that was in the other class. Different viral strains can therefore infect the same individual. That's why with COVID-19, some people have been infected 5 or 6 different times. It's because the virus is changing, mutating, and then the new strain can infect the same individual. It is also true that with COVID-19, your immune system isn't responding very strongly when it infects you normally. For some reason, it's a much stronger response if you get the vaccine. But the point is the virus can change. The best example of that is the flu virus. And they say that this one changes rapidly enough that after you get a flu shot, your immunity decreases to a level that's not giving you any protection. And that's a little bit of an oversimplification. When you first get a vaccine, even if you're a young adult or a teenager, the immunity is never 100%. It's high, it's of over 90%. So let's say around 95%. And then with time, that immunity decreases. In the case of the flu shot, 3 years after getting the flu shot, it drops below a threshold number where they say you're no longer essentially protected anymore. In reality, you do have a little bit of protection, but with more time, it would drop to 0. And I don't know what that threshold is. It might be 50%. It could be 37% when they say you really need to get another flu shot. I don't know what that threshold number is. But the point is, about 3 years after getting the flu shot, your immunity will drop below this threshold. And then they say you're no longer protected. And as I stated, the immunity really drops like that. And so it would be longer than 3 years, but this would be very weak immunity down here.
Dr. Volpe,
Uh-huh.
we did get much further than this in our last session. So we had gotten to DNA virus multiplication.
All right. I thought this sounded familiar, but like I said, I teach this on Monday, Wednesday class too. So, all right, let's move on because obviously we're not in the right place. Let me see if I just didn't remove that to here button. And I've got another one in here. Yeah, see, we were supposed to be talking about RNA viruses. All right, thank you. That'll save some time. So I talked about there were 15 families of RNA viruses that infect humans and cause disease. There are more RNA viruses, more families, but they don't cause disease, at least that we know of, in humans. And this is showing you some different RNA viruses, different shape sizes. Multiplication of RNA viruses in more detail. I wonder where that 2 here goes to. Essentially, the RNA viruses replicate similar to DNA viruses. In fact, the double-stranded RNA separates into single strand, and then you use each strand of RNA as a template to make a new strand of RNA. But there are differences because it's RNA instead of DNA. And then the viral RNA replication happens in the cytoplasm. When we're talking about RNA viruses, it's important to note that they can be positive single-strand RNA virus when we're talking about the single-strand RNA viruses. That is the strand of RNA that can be read by the ribosome. So it'll code AUG, which will start the protein translation and also insert the amino acid methionine. The negative strand, antisense RNA, single-strand RNA, cannot be read by the ribosome, so it will not be translated into protein. All right, so when we're talking about DNA, you just transcribe it to make viral messenger RNA and translate that into making viral protein. If we're talking about RNA virus, if it's double-stranded RNA, it's very similar to DNA. One strand can be read as messenger RNA, the other strand cannot be, meaning the positive single-strand RNA. And it can be translated into viral protein. If it's single-strand RNA, the positive single-strand RNA can be read as messenger RNA, read by the ribosome, and be translated into viral protein. How the positive single-strand RNA replicates is the positive single-strand RNA is used to make a template, or it's used as a template to make the negative strand RNA, and then the negative strand RNA is replicated to make more of the genomic strand RNA. Any question about any of that? All right, for the negative strand RNA, how it replicates is it replicates a positive strand RNA and then uses this to make more genomic negative strand RNA. And that's how you replicate negative strand RNA, single strand RNA. Remember, the negative strand RNA cannot be read by the ribosome, so it's not translated into protein. How the negative strand RNA will be translated into protein is it'll make a copy, which will be the positive strand RNA, and then that will be read as messenger RNA, viral messenger RNA, to translate the viral protein. Any question about any of that? All right, let me blow this slide up a little. So negative single-strand RNA will make positive single-strand RNA and then use that as a template to make more of the genomic negative single-strand RNA. And then only the positive single-strand RNA can be translated into the viral proteins, particularly the capsid proteins, which will then get together with the genomic RNA to make the new progeny virions. Any question about any of that? Now, for a positive single-strand RNA, it will make negative single-strand RNA to make more of the genomic positive single-strand RNA. And then the positive single-strand RNA will be translated into the viral proteins, which will then get together with the genomic RNA to make the new progeny virions. Any question about any of that? So let's look at viral multiplication in the big picture, and that is a virus that is a positive single-strand RNA virus will infect a host cell, and it has to attach and then be brought into the cell either by endocytosis or if it has a envelope by fusion of the envelope to the cell membrane. The RNA will be brought into the cytoplasm. Remember, RNA viruses generally replicate in the cytoplasm. That's a rule of thumb. There's only one exception, and that's retroviruses, and we'll talk about that in just a little bit. So this is positive single-strand RNA, so it can be immediately read by the ribosome to make the capsid proteins or the viral proteins. And then the positive single-strand RNA will make a copy, which is negative single-strand RNA. And then that negative single-strand RNA will be used as a template to make more of the genomic RNA, the positive single-strand RNA. And then the genomic RNA will get together with the viral proteins to make the complete virions. They'll self-assemble. The RNA will be stuffed into the head of the virus. And then this virus will either lyse the cell in the lytic life cycle, or it will bud out of the cell in the budding life cycle. And then the process starts again where a virus can infect another cell. Any question about any of that? All right, for the negative antisense strand RNA, it will infect a cell and then be brought into the cell by either endocytosis or if it has an envelope by fusion of the envelope to the cell membrane. The negative RNA strand cannot be read by the ribosome, so it has to first make the positive strand RNA, and then the positive strand, single-strand RNA will be used as a template to make more of the genomic RNA, the negative single-strand RNA. The positive RNA strand of RNA, single strand of RNA, will be used to translate the protein. And then the viral capsid proteins will get together with the genomic negative antisense single-strand RNA to make the new virions. And then once again, the virus will either lyse the cell to release the viruses in the lytic life cycle, or if the virus follows the budding life cycle, it will bud out of the cell. Any question about any of that? All right, let's talk about the family of retroviruses. This is an RNA virus, but it is different because it replicates in the nucleus, not in the cytoplasm. And you'll see why this virus is different in just a minute. The retroviruses include the lentiviruses that include HIV-1 and HIV-2. Both species of virus can infect people and cause AIDS. And then another lentivirus is feline leukemia virus, which causes leukemia in cats, which they call it kitty AIDS because the cat will just really waste away and their immune system will become very weak and they'll get recurrent infections. It's not because of true AIDS, it's because the leukemia is weakening the immune system. Retroviruses are unique in that they contain the gene for the enzyme reverse transcriptase. And actually inside the virus, a retrovirus, you can see at least one copy of reverse transcriptase. Where the heck is that? There it is right there. Inside the virus. Reverse transcriptase is unique because it can make DNA from RNA. Let me get the paint open. So retroviruses have a positive single-strand RNA. I should probably make this a little bigger. Oops, wrong one. And there's actually two strands of that. That's not terribly important. Let's get that a little closer. And so there's the positive single-strand RNA. In the presence of reverse transcriptase, let me make an arrow. And I'm just going to put RT for reverse transcriptase. The reverse transcriptase can make a single strand of DNA. And let me see if I can select this and just copy it. Did it? And then let's mark that, I guess. So this is single strand DNA. And then reverse transcriptase can work on the single strand DNA to make double strand DNA. Which of course will hydrogen bond in the way DNA normally does. Come on. All right, any question about that? Oops, that's the wrong one. Double-strand DNA. This double-strand DNA can then integrate into the host cell's chromosome. Let me just draw an arrow this way. So it moves into the nucleus. Come on. And then the viral DNA, let me make an X chromosome. I'll change the color here. Come on. Can insert itself. Well, it didn't change the color, did it? I tried. Can insert itself into the, or I should say, a spot in the DNA, which we'll call it a chromosome. And it doesn't have to be the same spot. In fact, in different cells, it can be in different, it is usually in different spots in the DNA. Can be different chromosomes, whatnot. Like that all blue. When this integrates into the host cell's DNA, it becomes a permanent fixture of that host cell, meaning that host cell will contain this retroviral DNA for its lifetime. And that's part of the reason why AIDS is a risk, is that even if you were to get it cured, like we have had some patients where they no longer seem to be making retroviruses, and you can't say that people are totally cured because they could have in only one cell in their body retrovirus integrated into their DNA. And at some point later in time, this retrovirus could begin replication. And then you'd have more retrovirus in the body infecting other cells and whatnot. So the disease could start again, for example. Any question about any of that? All right, how this replicates is the normal way, and that is the DNA is transcribed to make messenger RNA. Oops, wrong color, but that's all right. Switch that to black. And so we make positive strands, single strand RNA, which is the genomic RNA. And then this can be translated into viral protein. I'm just going to go VP for viral protein. V protein. And then the viral protein can get together with the genome. So that should be labeled, I guess. And that will form the new virions. Oops, I need an arrow here. Any question about that? And then the new virions will, well, they'll bud out of the cell. Oops, come on. I'm just going to go bud. Any question about any of that? And that's how the retroviruses replicate. And this is also why they have their replication in the nucleus. And that is they have a DNA intermediate in their replication. Any question about any of that? All right. And that shows you the importance of retrovirus, reverse transcriptase, in that it can make single-strand DNA from single-strand RNA. And then it can also make double-strand DNA from single-strand DNA. I think this part is not discussed in your book. So the reverse transcriptase is a gene on the on the viral RNA. as well as there's a reverse transcriptase protein inside the virus. So let's go through the multiplication of retroviruses. In the grand scheme of things, where a virus attaches to a host cell, it's brought into the cell either by fusion of the envelope to the cell membrane or by endocytosis. The viral RNA will then be made right there into viral DNA. And this is showing it in one step. In reality, the viral RNA will make a copy of single-strand DNA, and then the reverse transcriptase will make 2 copies of single-strand DNA using DNA as the template the second time. And then that viral DNA will integrate into the host DNA. Remember, we said yesterday or 2 days ago that we call that a provirus whenever the viral DNA integrates into the host cell's DNA. And then that retroviral DNA can transcribe, making viral RNA. And this will be positive single-strand RNA. So it is the genomic retroviral RNA. And it can be read by the ribosome to make viral proteins. And then the viral proteins will get together with the genomic RNA to make the new virions, and they will bud out of the cell. And that's how the retrovirus gets its envelope. Any question about any of that? All right. You don't need to know that retroviruses have 2 strands of positive single-strand RNA in their virus. You don't need to know that, but it is something of interest. All right. Let's talk about coronaviridie, the last family of viruses we're going to talk about. And I may in the future pull this out because coronaviruses, COVID-19 is no longer exactly new and people are getting used to it. In fact, when I go into the store nowadays, almost nobody is wearing a mask to prevent the spread of coronavirus. Coronaviruses were actually long known to humans before COVID-19 ever showed up first in 2019. And that's because coronaviruses cause the colds in humans. They're the second most common cause of the colds. And these cold viruses are particularly prevalent among children, especially young children. Coronaviruses are a single positive strand RNA virus. They do have an envelope. Besides causing colds and obviously COVID-19, coronavirus also caused SARS and MERS. These are 2 diseases similar to COVID-19, meaning they both cause severe acute respiratory syndrome. And that's what SARS actually stands for. And then MERS stands for Middle East Respiratory Syndrome. All 3 of these are lethal. The human cold is generally not. MERS has the highest mortality rate, where about 36% of people who get infected with MERS will die. Fortunately, MERS does not spread people to people. People who get MERS are people who spend a lot of time around camels. Apparently, MERS is a virus which is native, if that's the correct word, for camels. And people who spend a lot of time with camels can get infected with MERS. But fortunately, MERS doesn't spread people to people. And that's a good thing because the mortality rate is over 1/3. SARS has the next highest mortality rate. Its mortality rate is around 10%. And SARS actually popped its head in the human population twice. The first time humans ever saw it was 2003. I don't remember the next time it came into prevalence. It was sometime in the 2000s, but I think something like 2007, but don't quote me on that. It was a serious virus because about 10% of the people who got infected died. But one thing good about SARS as well as MERS is that there are no asymptomatic patients. So almost everybody who has SARS or MERS has symptoms. And so we know they're sick and you know to avoid them, you know to give them treatment. And so it will not spread beyond the people who are at contact to the people who are in contact with the patients who have SARS or MERS. Unlike COVID-19, where a large number of people are asymptomatic, and young adults as well as children and teenagers, about 50% of them are asymptomatic. And I don't know if I mentioned this before. You might remember that President Trump put on a quarantine into the United States, that nobody coming from China could come into the United States. And then at the airport, they were screening people coming into the United States, checking to see if they have a fever and if they're feeling ill, and then quarantining people who did. That didn't keep COVID-19 out of the United States because so many people were asymptomatic. And of course, back then, when it first was around, we didn't know how many people were asymptomatic. Although I think they suspected that there were asymptomatic people. Because one of the first cases in the United States was a teenager. I think he was in Seattle, but he was definitely in Washington. And he had not been around anyone who came from China, not been around anyone who was sick with COVID-19 that he knew of. And yet he had COVID-19. And so the health authorities guessed that COVID-19 was circulating in the United States and probably was asymptomatic. So they guessed that pretty early, but they didn't know how many people were asymptomatic. And we didn't know that until much later, at least 6 months later, maybe a year later, when they did vast epidemiological studies. And then they found a lot of people were carrying COVID-19 and had no signs or symptoms of it. COVID-19 initially had a mortality rate between 0.2% to about 3%. That mortality rate has reduced to about half that now. And we'll talk a little bit why that reduction has, but the strains that have been going around ever since the virus Omicron, that variant of COVID-19, has had a lower mortality rate. And then COVID-19 spreads more easily than the flu, with each person who gets infected likely to infect between 1 and 2.5 other people. And that's why COVID-19 keeps continuously spreading in the human population. Not to mention that it does change its antigens so that the immune system, even if you are immune to it, you can still get infected by a new strain. And there have been some people who have been recorded as getting the COVID-19 6 different times. Anyone know of anyone who's gotten it 6 different times? Nope. The highest I know is somebody who's gotten it 3 times, but beyond that. don't know anyone who's gotten it higher than that. All right, COVID-19 does have a different name. Its scientific name is Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. This name is so similar to SARS that even scientists don't like using it and frequently will use COVID-19. COVID-19 actually is an acronym for when the virus was found. So it's called CO for coronavirus, or I guess COV for coronavirus. And then it was identified, ID, in 2019, and that's where the ID 19 comes from. Any question about any of that? It is closely related to bat coronaviruses and is likely to have originated in bats. However, this virus is a recombination of two different bat coronaviruses and a third coronavirus that infected pangolins. So of three different coronaviruses recombined to make COVID-19. The spikes on COVID-19 are actually coded for the pangolin coronavirus. And it's these spikes which make COVID-19 so infectious among humans. It's likely that an intermediate brought the coronavirus from bats to humans. And they think that intermediate was actually a pangolin. And if you don't know what a pangolin is, it's a spiny anteater. They believe that at a Chinese city, I think it's Wuhan, something like that, that they had in the animal market, sort of a giant flea market where they're selling different animals for people to buy and then consume. They had pangolins as well as other animals. And it's believed that an animal in the market brought the COVID-19 to an individual, and then that individual picked up COVID-19, got it, and then spread it to other people. And then it got in the human population. That's all I'm going to say on that. It's likely the person who either bought the pangolin got infected, or maybe the person who was selling the pangolins got infected. But we don't know who the first patient was who got COVID-19. We do know what city it came from. And we do know that it came from an animal market. What do you call that? Where you sell animals at a market? It's not really a flea market, but something like that. The Chinese have now realized that when they sell exotic animals, when there's lots of animals in the place and lots of people in the place, that it's very likely that a virus from the animal can jump to a person. And they know this because not only was it witnessed in COVID-19, but twice SARS came from a flea market in China, from this animal market. And that's why SARS came from China as well as COVID-19. And the Chinese have realized, geez, we've had 3 events now in the last, well, it's less than 20 years, where a lethal virus has jumped from animals into humans and then began spreading among humans. And they said, maybe we should regulate these markets more and get rid of all these exotic animals, because 3 times in less than 20 years is a great number for a lethal virus to be introduced into the human population. So hopefully we'll have fewer events like this in the future. But like I said, in the last 20 years, it's happened at least 3 times twice with SARS, and then once with COVID-19. Any question about any of that? How we know about let me back up a minute, that the coronavirus was a recombination is that they sequenced COVID-19 and then they recognize where the coronavirus pieces came from. The transmission of COVID-19 is mostly via droplets from coughs or sneezes within a range of 6 feet, although it can be spread by breathing, talking, or especially singing. Singing tends to spread COVID-19. It may spread further than 6 feet, so it doesn't have to be only 6 feet. You can get it just walking into the room, for example. It can also spread via contaminated surfaces, meaning where the virus will get in a droplet and the droplet will then, I don't know, go on the table, and then you can pick it up on your hands and then inoculate a mucous membrane in your face. It can spread that way, but for some reason, COVID-19 does not tend to spread by indirect contact from a contaminated surface. It can, but it generally does not. It tends to spread directly person to person. It's usually breathed in. It is a virus that can survive for 3 days on plastic or metal surface, or a day on paper or cardboard. Any question about that? And in the old days, I used to hold my mail for at least a day so that the COVID-19 that might be on it would die. The symptoms of COVID-19 is it's mostly flu-like. And actually the later variants of COVID-19 have been even more flu-like, where the runny nose and the sneezing are very common. The shortness and breath are more common in COVID-19 than the flu, but the later strains are not as common, the shortness of breath and deaths, as in the earlier strains. Mortality of COVID-19 increases with age, meaning the older you are, the more likely you are to die of COVID-19. The ill health of the patient, meaning the iller, the more sickly you are, the more likely you're to die. Men are more likely to die than women, and Blacks and Hispanics are more likely to die than whites or Asians. The reason for some of these are not well known. Like we've known for Blacks and Hispanics now for several years, and it's been verified in the United States and Mexico and Canada, but we don't know why Blacks and Hispanics are at greater risk of dying. And you can't say it's just because Mexicans have a lower standard of health than Americans, because you can have Hispanics in the United States who are very upper class, meaning they're in the American lifestyle, or in Canada, and they're more likely to die than a white or Hispanic, a white or a Asian, sorry. Men, it's well known that men are at greater risk for many diseases, and they're more likely to die than women. It's a little surprising that COVID-19 is more likely to be more severe in men and more likely to kill men than women. The incubation period for COVID-19 is 2 to 14 days from initial exposure to getting the first signs or symptoms. However, you have to realize that a large percentage of the population is asymptomatic. So they'll get the virus and it will be growing in them, but they will be asymptomatic and they can spread the virus to other people. The actual death rate of COVID-19 in the United States, and this is about a year old now, was 1.8% of people who got infected with COVID-19 died. and that's for the total time since COVID-19 started infecting Americans. Like I said, was more lethal in the past than it is today. The most lethal variant was actually the Delta variant, which, if you don't remember, that was the variant that struck before Omicron. For prevention, use correct hand washing, keep your distance from people, especially when they're coughing, and then don't touch one's face. If you're sick, you should wear a mask. It's best to stay home and cough into your elbow or your tissue. Oh, here's one other thing. If you'll notice, the symbol I chose for my class is actually coronavirus. I forgot to mention that. I just saw it here and said, I actually chose this symbol for microbiology like 10 years ago, or maybe even longer. This isn't COVID-19. This is a human coronavirus that causes the human colds. Okay. But it is a coronavirus. And it's very appropriate after COVID-19 to have that simple symbol for microbiology. All right. Any question about COVID-19? Any question at all? In the early days, they tend to give the virus names, like where there was the alpha variant, the beta variant, the delta variant, and then Omicron. I don't know if that's a Greek letter or not, Omicron. But after Omicron, they started calling them by letters and numbers. Although there was one variant that they did have both letters and numbers as well as a name. I don't remember what that variant was, but it's a variant that was fairly recent. It's not, I think, the current variant spreading, but the variant before that, or the second variant before that. So maybe 6 months ago, they had one that had a name. Any question about coronavirus, COVID-19? If not, let's talk a little bit about viruses and cancer. Some human cancers are caused by viruses, and this is about 10 to 15% of human cancers. For example, human papillomavirus is known to cause cervical cancer and skin cancer. Hepatitis B virus and hepatitis C virus is known to cause liver cancer. So some cancers are definitely caused by virus, or the virus plays a role in making the cancer. That isn't true for all cancers. Like Tang found that breast cancer and glioblastoma have no association with any virus infection and the cancer. So not all cancers are associated with viruses, but 10 to 15% of human cancers are. Let me say, I'm not going to talk about this. Cancer is uncontrolled growth of cells. These are your own cells that are growing uncontrollably, causing the cancer. We're not going to talk any further about cancer in that way, but I do want to talk about the ways that viruses may cause cancer. First, you can have a virus integrating into the host cell's DNA. And that's when we call the formation of a provirus. It permanently makes a genetic change in the cell, meaning once that viral genome integrates into the host chromosome, that will stay that way in that cell for the lifetime of that cell. That can change the expression of the genes in the cell, as well as mutate genes in the cell. Imagine you had a tumor suppressor gene. If you don't know, those genes do exist. And let's say a virus integrated right in the middle of that tumor suppressor gene. the tumor suppressor gene is going to be knocked out. It's no longer going to be a gene product made from it, meaning the gene will be turned off because the DNA is now messed up. So a provirus can actually mutate the DNA, leading to cancer. And I mentioned that retroviruses obviously integrate into the host cell's DNA, but some of the DNA family of viruses also integrate into the host cell's DNA. Any question about any of that? All right, the second way a virus can cause cancer is the virus carries an oncogene. An oncogene is a gene that is mutated, but the gene is involved in cellular growth. So imagine you have a gene that causes the cell to grow. Now, normally this gene will be under control by the cell, so it would only be expressed when it's needed, and the cell would only grow when it is needed. Like when the child is obviously growing, this gene may be expressed to have the cells divide and grow. Now, let's say we mutate this gene, and now the regulation of the growth is lost, and now the cell just keeps on growing. This is what we call an oncogene, a mutated form of a normal cellular gene involved in cell growth. And so this oncogene, when it gets in a cell, will cause the cell to grow uncontrollably. And there are viruses that carry an oncogene. And they can cause tumors because when the virus infects the cell can now lose its control and just start growing uncontrollably. Any question about any of that? We won't talk about why a virus may have an oncogene, but the virus, when it infects a cell, it wouldn't mind if that cell were to replicate and make more cells, because obviously that cell has the virus in it. All right, let's move on. So there are two ways that viruses can cause cancer. They can integrate into the host cell's DNA, mucking up the host cell's DNA, maybe even mutating the host cell's DNA. And then the second way is you can have a virus bring an oncogene into the cell. And then that oncogene will then make the cell grow uncontrollably, leading to cancer. All right, I mentioned that there was another life cycle of viruses in humans and actually any animal, and that's the latent life cycle. That is where a latent viral infection has occurred and the virus has gone into latency. When the virus is in latency, the virus is asymptomatic, and it may remain asymptomatic in the host cell for long periods of time. The cell is just in stasis, or not the cell, the virus is in stasis. When the virus is in latency, the virus is not replicating, is not causing disease. The problem is that when the virus leaves latency, the virus can then replicate and then start causing disease. So when the virus is in latency, it doesn't cause disease. A latent virus is usually a budding virus. And while the virus is budding, it's obviously reproducing and causing disease. But then the budding virus can leave the budding life cycle and enter the latent life cycle. The virus is not really much metabolically active. It is not replicating. It is not causing disease. It's just, I don't know, in stasis, sitting there, waiting for a time to come out. Two examples of a virus that are latent viruses are the cold sores. When the cold sore goes away and the virus is now in latency and it's just waiting for the virus to reactivate, in which case you'll get another cold sore. There usually is a trigger for getting a cold sore, and of course it's getting a cold. Shingles is another latent virus. When the child has chickenpox, the virus is in the budding life cycle. It is actively reproducing and causing disease. The virus can then enter the latent life cycle, and generally it just sits for years, maybe even decades. And then when the child is now an elderly adult, the virus can come out of latency, re-enter the budding life cycle, re-enter the multiplication of the virus, and cause disease once again. And that disease is always the shingles. The first time you get the virus, it causes the chickenpox. All later times, it causes the shingles. The trigger for the shingles is a stress, and usually it's the old age of the patient where the immune system drops in the patient. And then the patient is no longer protected from the chickenpox. And then they get shingles. Any question about any of that? All right, so that's the latent viral life cycle. If you remember, I did mention there is a persistent viral life cycle. It's very similar to the budding life cycle. The difference is it's a naked virus and it leaves the cell in a vesicle. That's why it's a naked virus. But it is similar in most respects to a budding virus because very few of the virus typically are reproduced. They leave the, well, inside the cell, they're surrounded by an envelope, which is the vesicle membrane. And then the vesicle will take it to the cell membrane, fuse with it, and then dump the virus outside the cell. That is a persistent viral life cycle. Let's talk a little bit about a persistent viral infection. A persistent viral infection is a virus that persists over a long period of time. Sometimes the virus will just increase in numbers over a long period of time, and it never decreases. And the signs or symptoms start mild, and with time, they get just worse and worse. Sometimes the virus, persistent viral infection, starts slow, builds up, and then it gets better, and then it actually just disappears. An example of that would be, what is that disease called? The kissing disease, mononucleosis, where the patient becomes infected, they enter an incubation period, they then enter a prodromal period, which is fairly long, and then they enter an illness period, and it lasts for a long period of time, at least 3 months, or is it at least a month? I forget, at least a month, maybe 3 months. And the patient just gets sicker and sicker, and they get weaker and tireder. But it does get better. You do get better. You do enter a decline phase. You do enter a convalescence phase. And then the virus will eventually be thrown off. Mononucleosis. That isn't true for AIDS. In the old days, before modern medicine, HIV would likely eventually cause AIDS, and then the patient would just get sicker and sicker until they died. And that was a persistent viral infection. All right. So the persistent viral infection can be fatal, doesn't have to be. HIV can be fatal. With modern medicine now, this disease can be managed. And so death is not certain, although the patients, even with modern medicine, do not live a normal lifespan, but they're close to it. But many of the persistent viral infections can be tolerated. Like some people in the United States get infected with cytomegalovirus or CMV, and they just tolerate it. It does get worse with time. So let me end this. I'll do it on paint. I can get it here. So with cytomegalovirus, it does get worse with time, but then it plateaus out, doesn't get any worse. Okay. And then the patient just tolerates having virus nearly constantly replicating in their system. So that can happen. Obviously, if it were to get worse and worse, it would eventually end because the patient would eventually die. And that's what happened with AIDS, where the viral load would increase rapidly and then actually should back that up just a little bit. It doesn't decrease. There we go. And then the patient dies, and so there's no more part of the graph. And like I said, if that were to happen with cytomegalovirus, it eventually would get worse and worse, and then the patient would die. And it doesn't. Like I said, it just keeps producing at about the same number, but doesn't get worse. So after a while, it reaches a plateau. The chicken pox can be a virus which causes a persistent infection. In my mother, she got shingles, and for the last 3 years of her life, she had persistent shingles, where the shingles never went away. It might have even been more than 3 years. It might have been 5 years. Any question about any of that? Oh, excuse me. So let's look at the different viral infections. You can have an acute viral infection where the patient first becomes exposed, and then very rapidly, the number of virions increase in the patient, causing an acute infection, but then very rapidly, the virus decreases and then disappears. An example of this acute infection would be the human cold, where the virus hits, increases in number very rapidly, and then decreases very rapidly, and then disappears. So the virus runs its time very rapidly. The human cold is in a case of an acute viral infection. A persistent viral infection, which is shown here, would be HIV before the modern medicine, and that is the virus would get infected in the patient and they wouldn't even know they're sick because the virus is so low. And then the virus numbers would increase, 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 and then bloom. That would usually be when the patient got AIDS, and then the graph ends because the patient dies. Now, like I said, in some cases, like cytomegalovirus, do I still have that there? Yeah, I do. The virus load would increase over a time, but then it plateaus out, and then the body is constantly shedding a certain number, but the virus is tolerated by the patient. And then there's a latent infection where the patient first gets an acute infection and that would be something like chickenpox. And then the virus is actually down here at 0. Okay. It could be a long period of time, like for a child. They're going to go through childhood, go through adulthood, and not until they're elderly do they have the latent infection causing shingles, where the virus leaves the latent life cycle and re-enters the budding life cycle. And so the patient is only sick in 2 points, this one here and this one here. Any question about any of that? All right, if not, let's talk about prions. Prions are proteinaceous infectious particles where a protein is infectious and the protein causes disease. There are some inherited cases, but the prions can be transmissible by ingestion, organ transplant, and by surgical instruments, bringing the prion protein into the patient. Prions cause spongiform encephalopathy. This is vacuoles in the brain in both humans and other animals. Let me show you a picture of that. There we go. This is a normal brain here. These are all prion brains. Do you see why it's called spongiform encephalopathy? Because the brain looks sort of like a sponge. In animals, the best known example is bovine spongiform encephalopathy, which is more commonly called mad cow disease. But it also causes scabies in sheep and cervid chronic wasting disease in cervids. In humans, we don't call it any of these names. We don't even call it spongiform encephalopathy. It's called Creutzfeldt-Jakob disease after 2 doctors who noticed the disease in certain families, meaning they were inheriting a gene which later caused Creutzfeldt-Jakob disease. And this disease only manifested in late middle age or elderly lifespan. So the patients, when they were young, were reproducing, and then the gene was continuing in the families. And then Gertzmann-Straussler-Schnenker syndrome, and I could be butchering this, I'm not German. They were following different families, which were a different inherited gene. It's the same gene, but a different variant of it. And they were following that. And we'll talk a little bit about the inherited syndromes, but and then another case of humans is called fatal familial insomnia, and then Kuru. Kuru is the one I know the best, so I'll talk a little bit about it. Kuru is a disease that was seen in the Fore tribe of Papua New Guinea. And that's this red area here in Papua New Guinea. It was spreading in the Fore tribe because the Fore tribe had an unusual funeral ritual where the people would cook their relatives and then eat their dead as part of the funeral ritual. And because of that, some of them begot the Kuru, a disease causing the patient to be unable to walk, eventually loss of the ability to swallow or chew, drastic weight loss, and then would lead to death. Surprisingly, the mothers in the Fore tribe noticed their children getting sick of Kuru, and they suspected that it was from eating their own dead relatives, especially a dead relative who died of Kuru. And so the mothers themselves in the Fore tribe stopped eating their dead and then refused their children from eating the dead. However, Kuru was continuing in the Four tribe because the men were still eating their dead relatives. And the missionaries came in to Papua New Guinea and saw the people, at least the men, eating their dead relatives. And the missionaries were appalled at this. And they just said, this is anathema. We cannot allow it. We have to put a stop to it. And they actually did. They passed laws saying that you can no longer eat your dead relatives in the funeral. And I guess there were punishments if you broke the law. And eventually, because of the missionaries' activity, Kuru became extinct in the Four tribe. So you can say what you will about missionaries and how they're pushing on their beliefs to the natives, and generally they harm the natives. But in this case, in my opinion, they actually did the Four tribe a favor. They got rid of the Kuru from the tribe. Any question about any of that? All right, I've already talked about that. How Kuru, as well as all prions, replicate is there is a normal cell gene in our cells, which is particularly important in the nerve cells, and it's PRP superscript C for the normal gene, and it codes for a cell surface protein. This gene is important in a normal nerve cell function. There was also another variant called PRP superscript SC, SC standing for the Scrapes protein. And these two proteins have the same amino acid sequence. The only difference between the protein is the folding pattern differs between PRPC and PRP superscript SC. So these two proteins are the same amino acid sequence, they just are in a different folding pattern. The PRP-SC protein can come up to the PRPC protein and then change the folding pattern of the PRPC protein to change it into the PRP-SC Scrapes protein. And that's how this protein replicates. Comes up to the normal protein and then changes its folding pattern to the Scrapes protein. The cell says, oops, I don't have enough PRP superscript C, so I'll make more of it. And then they make more of it. The trouble is now this cell has two copies of the PRP Scrapes protein. And so one of them is going to more likely come up to the PRPC protein and then change it into the Scrapes protein. The cell is going to say, once again, whoops, I need more PRPC protein. And so it'll transcribe more of it, transcribe and then translate more of it. And this is how the PRP Scrapes protein replicates. And the PRP-SC, the Scrapes protein, will accumulate in the brain cells, eventually forming plaques and eventually causing the cell to die. Obviously, plaques in the brain is not a good thing, and then having the brain cell die is not a good thing. And that's what gives rise to the prion disease. Any question about any of this? This is an unusual way of getting a disease where the protein causes the disease, and how the protein replicates is it comes up to its normal form of the protein and then causes the normal form to unfold into the Scrapes or infectious form. Any question about any of that? Mad cow disease was a problem in Great Britain. I don't remember when, in the early 1990s or sometime in the 1990s, I think. And the problem was because the British were feeding cow brains to cows. They wanted an economic benefit of the cow brain, and they thought just throwing it away was not of economic importance and wasteful. So they ground up the cow brains and added it to grain, and then fed that cow brain tainted grain to cows. And the trouble was there was a scabies, a cow with the scabies protein, the prion, and their brain was ground up, given to the grain. The cows ate the grain, more cows got scabies, and you can see how this would balloon out of control. The British actually solved it by incinerating all of their cows and then buying new cows from out of the country and importing them to Great Britain. Drastic measure, but that's what they had to do to get rid of it. And I should say that besides the cattle getting mad cow disease, people and British cats were also becoming infected with the prion disease. About 50 British people came down with prion disease at the time, and about 50 British cats, both household cats and zoological cats, came down with prions. Any question about any of that? If not, let's talk about viruses and viroids. Plant viruses resemble animal virus in many ways. They do cause diseases in plants, and some of them are economically important, like important crops like beans, corn, sugarcane, and potatoes. They can all get plant viruses. Because of the tough cell wall, plant viruses tend to enter plants through a wound or a parasite. And that can be either a nematode, a fungus, or an insect, can damage the plant, and then the virus can then infect the plant cell. Infected plants can spread the virus in their pollen or their seeds, and that's how the plant virus spreads. Plant viruses do not cause disease in humans or in any animals for that matter. That's why we call them plant viruses. They infect plants, kingdom plants. And so we're not going to talk about that further in this class. Any questions about any of that? If not, let's talk about viroids. Viroids are a naked RNA molecule. So you can think of this as an RNA virus minus the protein code. This naked RNA causes disease in the cell that it infects. Fortunately, viroids are only plant pathogens. There are no known viroids in animal cells. So all viroids infect plant cells, and it's just an RNA molecule, naked RNA molecule. When it gets inside the cell, it causes disease in the plants. In potatoes, it causes spindle tuber viroid, which causes the potatoes to be stunted. So its importance is economic importance, but it does not cause, viroids do not cause human disease. Any question about any of this? Any question about chapter 13? If not, I will be in the lab for 15 minutes, starting at 7. After that, I'm going to log out, but I'm only there to answer questions. If you don't have any questions, you do not need to go to the lab. All right, I will say goodbye, unless there's a question.
Thank you.
All right.